Okay, so what do you remember about incipient plasmolysis? Grace, what have you just read there? What does it say?
It's the point when the cell membrane is just touching the cell wall.
Yeah, so incipient plasmolysis, the cell membrane is just touching the cell wall. So cell wall, cell membrane just about touching it and no more. What about the pressure? Zero. So pressure potential is 0. So the equation, the water potential of the cell is equal to the sum of the solute potential plus the pressure potential. But we're saying at incipient plasmolysis, that is 0. So if we can work out the water potential of the cell, we know that it is the same value as solute potential. The two figures are the same because there is no pressure at incipient plasmolysis. Now, we've got some red onion at the front there. I've got different concentrations of sucrose solution. So if you make a note of the concentrations, so we've got, these are molar. So units are capital M for molar. So we've got 0.0, which is obviously pure water. We've got, well, don't need to put 0. So 0.0, 0.2, 0.4, 0.6, 0.8, 1.0. So we've got pure water and then 5 other concentrations. So if you make a note of those. What we're going to do is we're going to cut sections of the onion epidermis. So I'll show you, it's just a very fine layer. Whenever you looked at onion cells in 4th year and in 1st year, you've done this. So we're going to cut bits of onion epidermis. But we want to put some into each of these concentrations here. So you need at least 6 little squares. Some books would say, 1 centimetre by a centimetre or half centimetre by half centimetre. It doesn't need to be even as big as that, but try and get it about half a centimetre by half centimetre or bigger. So you want to cut a few squares. You want to cut at least 6 because you're going to put 1 into each concentration eventually. So you want 6 squares, aiming for half centimetre to centimetre square. But you're going to put them into pure water first of all right? So we're just going to work in groups of 4, I think 4 or 5 is the easiest thing, all right? So we'll just have 2 groups, all right? So we have 1 group here, 5, and 1 group here, 6, is that all right? No, that's 5 over there. So each group is going to need, well, I would say, try and cut 12 squares. So you've got 1 to use and 1 in case it goes wrong, okay? Cut 12 squares, all right, for your whole group. And you're going to put them into a Petri dish which has just got pure water in it. And we'll leave them there for a few minutes because we want all of the cells to start off being turgid. And that's the first thing you're going to do. So you will need that. You will also need 6 little dishes. So we've got Petri dishes there. And you need to, we're not going to bother putting a label on it because it's messy to get it off. Just get a little piece of paper and write on it, 0.0, 0.2, 0.4, 0.6, 0.8, 1.0. And pour some of the solution into it. It doesn't actually matter how much. So long as there's enough for you to be able to put your, later on, you're just going to put your epidermis in there. Okay, so, a few millimetres deep is plenty. You don't need to measure it. So somebody else can be, getting these already, pouring the solution in there. But for now, all of your 12 pieces of onion epidermis are going to go into pure water. So pure water is your first one there, 0.00 molar is pure water. So for now, I'll show you how to get the onion epidermis, how you're going to cut it. We need the 6 Petri dishes, or there's evaporating dishes there would do the same job. It doesn't really matter. You need 6 of those. Pour the solutions different concentrations into each one. Make sure you've got a piece of paper labelling it. And later on, then we'll use those other ones. Okay? So we'll pause that now. Okay, so we said put your cells into pure water first of all. We would expect onion cells are put into pure water. Well, pure water, so outside, the pure water has a water potential of 0. Inside, the water potential of the cell is always going to be negative because there's sugars and salts there. So 0 is higher than a negative figure, so you'd expect water to move in. So you'd expect the cell membrane to be right up against there and the cells to be turgid. And in these red onion cells, the pigment will be quite clear, so it's a lot easier to see. So when I looked at my cells that were put in pure water, okay, and low power magnification, that's what you look at. First of all, you can see lots of cells. Now, whenever you were peeling off the epidermis, you noticed that it wasn't all, it was sort of a bit stripy. Patches were pink. So what you're just going to do, we're just going to count the pink cells. We're going to ignore the clear ones because they're too hard to see the contents of. Now, don't panic because there's far too many cells there for you to count. So you focus under the low power, first of all, to actually get your cells in the right place. Make sure you've got some pink cells there in the centre of your field of view. And then you move to the medium power. Now, I find it very difficult to get a photograph. You need to get a photograph, but it doesn't matter. So part of that is in focus, part of it is blurred. That is okay. It's just evidence that you've done it. Don't panic. All right? So what you would do is count how many pink cells there are there altogether. And of those pink cells, how many are plasmolyzed? Well, plasmolyzed, anything that is pulling away from the cell wall is plasmolyzed. Right there, all of those cells are right up against. Now, if we zoom in there, you can see that the clear bit around there, so this, if I just point here, the clear bits there, that's the cell wall, those are cell walls. So you can see clear bits, but that's fine because the pink goes right up to there. I'll show you some plasmolyzed cells later on. You would expect all of those cells to be turgid. So count how many cells there are in your field of view. You might need to estimate, okay? So I am not going to come around and double check whether you got 24 or 25, all right? But look, count how many cells you got there altogether. Note how many are plasmolyzed, which you would expect to be 0, all right? And then when you've done that, you can start, everyone then can start to make up the slides with the other concentrations, and that's what you will do there. Okay, I'll show you a plasmolyzed cell here in a moment, all right? One there, they were like pretty full. Okay, what I've done here is take some plasmolyzed cells just to show you what they look like. Now, I mean, my photograph, I can't see the whole field of view there on the camera. That's fine, that is fine. You just need at least one photograph of cells. Shh. Okay, so if we look at that, you can see that there's a definite difference between the cells you looked at in the previous. As over here, you can see that, you know, if this, you imagine, you can't really see the cell wall, but you imagine the cell wall is there. And there's these big gaps here. So those cells are actually all plasmolyzed. Now, you can't tell just by looking at it whether cells are at incipient plasmolysis or not. And so what do we use? What did we say last day? What figure did we look for? 50%. So what you're going to do, you're going to count the number of cells, count the number of plasmolyzed at 0, 0.2, 0.4, 0.6, 0.8, okay, and 1. And then we'll take our figures and draw a graph. We'll do that afterwards. So right now, what you need to do, if everybody takes a count from that first microscope, okay, and then after lunch, as soon as you come in, we'll let our cells sit in the solution for a while. And then as soon as you come in after lunch, you're going to set up. Each of you is going to have another slide to set up of a different concentration. And you can set that slide up. You can take your figures. Then you can go to somebody else's microscope, take a count there, and so on. Okay? Do you understand? Yep, okay.